All right, ladies and gentlemen, welcome back. I believe this is the fourth video for chapter two. Uh, so you should have had vocab, you should have had properties of water, you should have had macromolecules part one, and this is now going to be macromolecules part two. Last time we talked about how you take a bunch of atoms, put them together to make a molecule, we call a monomer, and you get those monomers together and you make a polymer. And you get a really big polymer, we call it a macromolecule. Today, we are going to be going into the details about those macromolecules. We're going to talk about all four, hopefully we'll talk about all four, double check that when we get in there, but at least three of them are food. The fourth one, nucleic acids, are not food, all right, that's your DNA, but carbohydrates, proteins, and lipids are all food-based. So let's go into that. We'll talk about the food chemistry of your body. All right, our first one, carbohydrates. Composition. What are carbohydrates made out of? Carbohydrates are made out of, this is the refresher, this was in the last video, monosaccharides. One monosaccharide is sugar. One sugar molecule. Those are your monomers. You get a bunch of them together, you get a polymer of sugars. Macromolecule we call carbohydrates. Those monosaccharides are made up of carbon, hydrogen, and oxygen. Now, do I care about which individual atoms are in there? Personally, no. I care that you know that there's carbon. However, the keystones, I have been informed, do want you to know the difference at this level between the macromolecules, between those monomers. So know that carbohydrates, carbon, hydrogen, and oxygen. That's super easy to remember. Check this out. Carbohydrate, carbon, water. That's right, carbon and water. Hydrogen and oxygen, H2O. Carbon and H2O. So easy to remember. This is going to be at a 1-to-1 ratio. Another thing that pops up in the keystones, 1-to-1 ratio, meaning one carbon, two hydrogens, one oxygen. Right? Okay, here there's a whole bunch of them, but for every one carbon you see, you are going to get two hydrogens and an oxygen. H2O, carbon and water. What's the structure look like? Well, they're going to be fairly flat. Right? Each one of these represents a monosaccharide, what we're going to call glucose, and they're going to link together in a fairly flat plane. So here, that's one monomer. That's two monomers linked together. We call it a polymer. And then four of them linked together is still a polymer. No matter how big you get, this is going to be a polymer, right? Even larger polymer, it's still just a polymer. Eventually, though, it's going to be so big that we are going to consider it a macromolecule. It's still just a polymer. But this macromolecule is your carbohydrate. And those carbohydrate chains can link to other chains and make these big sheets of carbohydrates. So they're very flat-ish. You know, think of this three-dimensionally, like laying down. This is like the top, and it's kind of be a crumbled but mostly flat plane. And it's very easy to break. You can snap them. No big deal there. That's why carbohydrates tend to be crunchier foods. Right. Function. What does a carbohydrate do? Well, think about crackers, think about pasta, think about breads, right? These are your high energy bonds. Remember, sugar, monosaccharide, one sugar. Sugar is for energy. So they're going to store energy in those bonds, and when you break those bonds, all that energy comes out. I showed that in that last video. I showed you this picture. Zooming in on that bond, see, we're right here where the hydrogen and the carbon meet. Zoom in on that, and if you pull those two apart, all that energy comes out, and that's the energy that you use to survive. And examples of carbohydrates. We talked about in the last video, there was a table where I told you guys you were responsible for filling in examples. So now it's time to check that out. Examples of carbohydrates, your monosaccharide can be glucose, and then a couple of polymers, we can include sucrose, fructose. Basically, anything that ends with ose, O-S-E, is going to be a sugar or a carbohydrate. So you probably heard of lactose, the stuff in milk that makes some people sick. It's a sugar, it's a carbohydrate. So those are your examples for that. Moving on to lipids. What are lipids? Remember, these are your fatty things. So composition, what's it made out of? Carbon and hydrogen. All right. There's some oxygen in there, but not nearly as much compared to the carbohydrates. So carbon and hydrogen. What's it look like? We typically draw it looking like a dot with two tails. What it really is a bunch of molecules up here and a bunch of different molecules down here lined up in a different certain ways. Weird thing about it is the molecules up here are polar, which means they have a charge, a positive and negative side, and they get along really well with water. But the tails coming down are nonpolar, which means they don't get along with water very well. So this is going to behave very strangely in a watery environment. And that's what your fat does. Your fats and oils, waxes, these things do not get along well with water. So what is the function of these lipids? Well, remember, they're your fats. There's your examples down here, fats, oils, waxes. These are things used to store energy. Right? You eat a lot of food, like we said in the last video. If you have too much, we're going to store a lot of the carbohydrates and a lot of the proteins in your lipids, and you build up fat. Another interesting thing for a future chapter is your lipids also make up your cell's membrane. So we already talked about cells. We should have already talked about that before. Your cells are storage units. Inside the cell are all of those organelles, and we keep them all inside, stored in the membrane, made up of these lipids. We again have a whole conversation about that in a different chapter. And proteins. Composition, what are they made out of? They are made out of amino acids. Carbon, hydrogen, oxygen, just like the carbohydrates, except we are now going to be adding nitrogen and sulfur to the mix. Right? This nitrogen and sulfur are going to add a little bit of flexibility. Right? I mean, these proteins are a bit more pliable. Where the carbohydrates were a flat sheet, the proteins are going to be bent into whatever shape they need to be in order to do their job. Right? Proteins are basically tools. And just like every tool in your toolbox, they are going to have a different shape in order to perform the job. So what's that structure going to look like? Whatever it needs to. Really, there is no one structure. Just like there is no one shape for all tools. Like, yeah, all different tools, they all have different shapes for different purposes. And that's your proteins. Yeah, they start off building up in a chain like the carbohydrates do, but because of that nitrogen and that sulfur, they start getting all twisted and wrapped around each other and wrapped around itself, and it's going to be in its own whatever shape. Usually proteins end up looking like a wad of chewed gum. What is the function of the protein? They form your body parts. Seriously, if I say name something made out of proteins, name literally any body part and it's made out of proteins. I mean, if you say hand, like that's not even that specific. You can name parts of your hand, fingers. You can say the skin. You can say the muscle, the bones, right? Blood. Everything is made out of proteins. They also control cell processes. Right? You have proteins in your cell membrane regulating what goes in and out of your cells because that's a tool, and all your body's tools are made of proteins. And they become enzymes, which is a different tool. More on enzymes in a bit. But long story short, there are tools that your body's going to use for chemistry, speeding up and slowing down reactions. So what are some examples of proteins? Like I already said, guys, name any body part at all. Muscles, skin, bones, enzymes, you name it, it's proteins. We're going to go back to enzymes in a bit, but nucleic acids, composition, what are they made out of? They are made out of the monomer nucleotides. You may have seen this before. Some of you have probably taken biology once before, and you would have remembered phosphate, sugar, and base. Phosphate, sugar, and here's the base. Right? There are 4 different bases, 5 if you count RNA, but whatever. So 5 different bases, and those are the letters of your genetic alphabet. Right? So the phosphate, sugar, and the base makes up one nucleotide, and what order you put them in, just like letters of the alphabet, you put them in a certain order, you can make a message, a code. And these are again made of carbon, hydrogen, and oxygen, just like before, and this time nitrogen and phosphorus as well. Yeah, there's the phosphate, there's your phosphorus in there, and nitrogen is in the nitrogenous base. And then your carbon, hydrogen, and oxygen is right here because look at sugar. There is a carbohydrate in your nucleotide. Because your nucleotide is so big that it has a whole nother macromolecule inside of it. So that's just a whole nother bag of worms that we're not really going to talk too much about at the moment. We'll go more into detail about this in a different chapter. Examples of nucleic acids. We are really going to talk about 2 of them. Oh, sorry, structure first. Structure is going to look like a ladder, a twisted ladder at your DNA. And function is to store and transmit hereditary information. Store, meaning all of these is just a very lazy way of drawing that nucleotide. This is just a series of nucleotides going all the way down, attached in a very specific order. So this is how they're stored, making a message. And by transmit, I mean you are going to make a copy of this and pass it on to your kid, so your kid is going to look a little bit like you. And examples, we only have 2, DNA and RNA. DNA is what you use to know what to look like. RNA is going to be used for the actual construction of your body. It's going to be telling the proteins what to do. Whole future chapter again later on. So back to enzymes. I set this aside because I wanted to talk about all the structures first. So now we got that out of the way, let's talk about these enzymes. Enzymes are proteins that are used in metabolic reactions. Oh, that's a weird word. Yeah, it's a keystone definition. All right. Metabolic has to do with your metabolism. A lot of people in here probably already know this, or some of you probably already know the word metabolism, right? You think it has to do with weight loss, and you're technically right, but it actually has to do with every chemical reaction in your body. That's your metabolism. And weight loss is a chemical reaction, so it's involved. Enzymes are used in those chemical reactions, or we call metabolic reactions. They are going to be used as a catalyst. In physical science or chemistry, you would have learned that word. A catalyst is anything in chemistry that is going to speed up a chemical reaction. It's going to make it happen faster, and it's going to make it happen easier. So you're going to use less energy to make this happen. Right? Enzymes are awesome like that. So I forgot I had those popping down. So metabolic reactions, any reaction in your body, and used as a catalyst, meaning they speed up reactions. How do they work? Well, they're going to work like puzzle pieces. Enzymes have an opening. So this purple thing represents your enzyme. Right? And notice that it has these specific shapes in here. In your body, they're not as easy to look at. This is a simplified version, so you understand it better. But it has these puzzle pieces. And this will only accept a chemical that has that shape. And this one will only accept a chemical that has this shape. So this is what we call the active site, the site of activity. The chemicals we're calling substrates are what's going into the active site. And the enzyme is going to help them change into something else. So these substrates would and could naturally fuse in your body without the enzyme, but the enzyme is going to make sure that they're together to fuse, to chemically react and become a bigger thing. And then when it's done, the enzyme lets go. And here's the thing, the substrates changed. It was two smaller monomers and became one big polymer, but the enzyme did not change. Right? The guy looks exactly the same way it looks before because it's not actually involved in the chemical reaction. It's a tool to make the chemical reaction happen. So think of your tools. If you were going to use a clamp to hold two pieces of wood together so that the glue can dry, when you're done, you take off the clamp and the clamp is still a clamp. The wood went from two smaller pieces to one bigger piece, but the clamp is still a clamp. And you can keep reusing that same clamp every time. You can keep reusing this enzyme every time. So if we're not going to run out of those enzymes, we're just going to keep reusing them. Well, sometimes we don't want to use it. So how do we stop it if we're able to keep recycling? We can provide something called an inhibitor. Okay? Inhibitors are anything that's going to stop the enzyme's activity. And there are some natural inhibitors in your body, painkillers, endorphins. Endorphins, people think endorphins are a chemical in your body that make you feel good. It's actually an inhibitor that blocks pain. So it's not that it's making you feel good, it's making you feel less bad. Endorphins are going to inhibit the chemicals that make you feel pain. Tylenol is the same way. You can introduce inhibitors with medicine. Take Tylenol, Advil, something like that. And we'll get two different kinds of inhibitors that are going to block these chemical reactions. One is called a competitive inhibitor, binds to the active site. Remember that active site where the substrate fits the active site, like a puzzle piece? A competitive inhibitor is competing with the substrate for that active site. So if it gets there first, then the substrate can't get in. That's why it's called a competitive inhibitor. They are racing for this spot, and if it gets there, no chemistry happens. Then we got non-competitive. It does not bind to the active site. Your non-competitive inhibitor is going to hold on to some other place on the enzyme. It's going to wedge itself in some place which alters the shape. And this is going to be very important for a future chapter. If your enzyme's shape changes, it cannot do its job. Look what happens here is I put this non-competitive inhibitor in the back, but that alters the shape of the whole enzyme, and now the active site got crunched in. So look at this. This opening goes straight up, boom. Goes straight up from this side, boom. It's too small. The substrate will not fit. So it doesn't have to compete for the active site. It's automatically going to win. So that's what inhibitors do. These are chemicals that are going to stop the substrates from fitting, and then no chemistry will happen. So that is it for the notes for Chapter 2. Take a look on Google Classroom, and I probably have left an assignment there for you. So good luck on that, and I will see you in the next chapter.